but it's my fault. I gave a key and I didn't put a point screen on it, so until this weekend they didn't get in touch with me. But they're working on it and they'll get it back as soon as they can. Okay, so we've been talking about gene expression, a little bit about regulation, and we talked about promoter elements, and we talked about the proximal promoter, and then we discussed distal promoter elements, which we call enhancers. And we talked about some of the properties of enhancers. We said that enhancers can be very long distances. They can be tens of thousands of kilobases away from that proximal promoter. They can essentially be flipped around in the DNA, it doesn't matter. They're far enough away that the protein binds things, they're functional. There are enhancer elements that go way beyond gene to the 3' end, the downstream side of the gene. There are actually enhancers that occur in intervening sequences within genes, particularly the 5S ribosomal RNA. And we said that these enhancer elements got their name because they increased expression, they enhanced expression. And I do so by enhancing the binding of polymerase to the proximal promoter. So the proximal polymerase binds to it, and the enhancer elements which bind to the enhancer somehow help to load it, which implies that they interact with the polymerase, and they do. I want to tell you a couple more things about enhancers before we go on. First of all, this slide simply shows that enhancer elements can be active, and more than one can work on the same gene. Because here's a gene with no transcription, you put one enhancer on, you get a single level, you put one in another, you get a couple of levels, whatever. But when there's two of them on there, they work synergistically and increase the transcription a lot. So these elements do work synergistically. Viruses use this. Viral elements are often very close together. These enhancer elements, which by the way, they were first defined as elements that activated transcription, but we now know that enhancer elements can bind protein that either activate or restrict or inhibit. So we have activators and we have repressors. And those elements sometimes can be competitive, they can be active, but they can also become competitive, which this essentially shows that if a repressor binds to a repressor region, that inhibits the activator. And this is very similar to what we saw right in the lactoc run, where if a protein bound to the operator, then when a repressor bound to the operator, that inhibits the polymerase from binding. Same thing here, when a repressor binds, it inhibits. Once again, these are cooperative. In this case, they can interact even when they don't interact. This activator can sort of be inhibited by binding to an inhibitor or repressor. And we have examples of all of these. I'm not going to tell you any of them. I said that they act by somehow increasing the binding to the promoter region. Some of these guys can react directly, as it shows here, but also, in many instances, there's another protein that's just an active thing. There's another protein complex, a large complex, that has probably 14 or 15 different proteins in there, and the complex is called the mediator complex. And it's called that because it mediates the interaction between the polymerase molecules, right? This is the promoter, there's the polymerase set there. So it mediates the interaction of the polymerase molecules with the different enhancer molecules that are found all around the chromosomes. So you can see that the enhancers interact with the mediator that interacts with the polymerase. All of these things act together to enhance or induce transcription or reverse transcription in a particular gene. So that's all I want to say about enhancers. Does anybody have any questions about enhancers? They're really sort of interesting. We study, like I said before, these enhancer elements are what confer tissue specificity on particular genes. So we have to have a protein that binds to a particular enhancer element, and if it does, then it's expressed in that particular tissue. Okay, so we talked about these enhancer elements, and you have to remember, or you should remember the thing. Now, when you talk about enhancer elements, you're talking about particular sequences in genes. Usually when we talk about an enhancer, we talk about a sequence in a gene. And that sequence is the sequence in that DNA that makes that region an enhancer region. none of these sequences work on their own or able to influence transcription. They all influence transcription by have binding proteins. And it's the proteins that bind to them that induce transcription. And there are several types of proteins or groups of proteins, not tons, but there are a few groups of proteins that are whose particular properties or particular characteristics are repeated or evolve over and over to affect transcription. And that's what I want to tell you about a little bit today, some of how these proteins are. One group of these proteins are called HTH proteins. And that stands for the helix-turn-helix proteins. And all of these proteins are characterized by having this red and blue particular thing. It's a helix, a turn, a helix-turn-helix protein. These proteins are most common. This is the most common type of protein structure is found in prokaryotic transcription factors. You can see here's the lambda probe, here's the lambda repressor we talked about, tryptophan repressor. A lot of these are found in bacterial cells. They all have this from helix to helix. The other thing you notice is in this diagram is that all of these are dimers. You can see here's one peptide, right, and it ends here. There's the other peptide that ends here. So they're two helix to helix proteins. They act as dimers. And there's a reason for that. One is or one result of that, or reason for that, I'm not sure which came first, but is that many times the regions of the DNA which we find are repeats. They're direct repeats. So you can see that if this helix binds one, this will bind another. I'll show you in a minute. On these helix-turn-helix proteins, one of the helices, this red guy, is involved in binding or interacting with DNA, whereas the other helix in the turn is more to localize, to determine exactly the orientation of the other helix, of the DNA binding helix. So the helix-turn-helix, this helix in turn, sort of orient those. And you notice that these are oriented sort of always in the same plane. Okay, so take a look at these. So here's a picture of the DNA. And you can see this is an area which has dyad symmetry. So the sequence this way and the sequence this way are the same, dyad symmetry. Here's this helix-turn-helix proteins. These are dimers. They've left out a lot of the proteins. They're only showing you the helix, the turn, and the helix. You can see the largest of the helix is the one that addresses the DNA. The other one is just maintaining the orientation so that these two helices fit into the DNA. Now, not all, but most DNA binding proteins, the proteins interact or address the DNA in the major group. Remember, DNA has a minor group, the distance between the helices is relatively small, and a major group where the distance between the helices or the wrap is rather large. And it's this large region where most of these interact. These proteins are homodimers. I don't know if they said that. I said that. So the sequence of this helix is exactly the sequence of this helix. And the sequence of the DNA going this way is the sequence of the DNA going that way. And they're binding around this dyad symmetry. Why do they do that? Why do they bind? Why do they bind as a dimer around? Why did this dyad symmetry evolve? And why do they bind around that dyad symmetry? Why do they bind as a dimer? All of those things influence the same thing. Why do they do that? It is recognition, yes, but what are you doing? You're recognizing, right? You're recognizing two of these things here. It increases specificity, right? If you have one of these and you're addressing a few nucleotides, then the specificity of this protein is very small. It's going to find those 4 nucleotides among the billions of base pairs over and over again in the genome. But when you have them in these first of all, you have this dyad axis symmetry between those, and that's much more unusual or much rarer to find those two sequences together like that with the dyad symmetry. When you have 2 of them, where you have a binding site for 2 of these helices, it's even more rare. So by having these deep dimers like that, and most of the DNA binding proteins are dimer, it makes the specificity of binding at that particular location much larger. Make sense? So this is the helix-turn-helix protein. It recognizes them. The second group are before I show you that, I just want to show you, the question is, so here I've said that they bind in this helix, right? They bind in this helix. They're recognizing a DNA sequence. They're recognizing that enhancer element of the DNA. How do they do that? Why does this protein bind here, not in this helix, not in that helix? How do they do it? And it has to do, once again, with the sequence of the nucleotides in that enhancer element, as well as the proteins themselves, the amino acids of the protein. And this just shows you two things. The proteins are able to address, identify enhancers, bind to enhancers, activate enhancers by forming hydrogen bonds. The recognition is mainly through hydrogen bond formation. This just shows a few. Here's the amino acid glutamine, and this is the base of the adenosine. And you notice that they're able to form hydrogen bonds. These arrows are very important. They show you the direction of the hydrogen in the hydrogen bond. So in other words, in this hydrogen bond, the donor is this hydrogen, and this one, the donor, is this hydrogen. Similarly, if you look at arginine, which has the amino groups on it, if you look at those amino groups, they're able to form hydrogen bonds with glutamine. Notice the direction of the bonds. The hydrogen bond is going this way on both of these. So if you were to take this arginine and to address this adenosine, it wouldn't hydrogen bond because these are going in the opposite, they don't go in the same directions, and vice versa. So there's specificity between the amino acids and the bases. And it's because of the arrangement or because of the side groups that are there, right, and the formation of hydrogen bonds. So it's the ability, it's almost like it's like complementarity between the amino acid and the base, just as it is base to base. So these guys address DNA in the major group primarily, although some do address the minor group, and they do it by the sequence of the amino acids. And this is just showing one, right? Remember, in those enhancer elements, if you look at those sequences, there's several nucleotides, and of course the amino acids in that helix, there's several of those amino acids which address in that minor group or major group. I'll show you that again in a minute. Okay, so now the second group. The second type are called